Hello everyone, good afternoon. First of all, I would like to introduce myself. My name is Dr. Waqasuddin and I am an assistant professor research at the Department of Pediatrics and Child Health, the Aga Khan University, Karachi, Pakistan. So my poster is all about the problem, you can say a global health problem of preterm birth. Let me share my screen with you. So basically, preterm birth continues to remain a major cause of neonatal morbidity and mortality, and around 50 million babies are born too soon or too soon every year. So we can say that 80% they are premature babies, or preterm birth occurs even in Asia, in just Asia, and in other sub-Saharan African countries. However, the problem is universal all over the world. So statistics from 2018 showed that Pakistan ranked Pakistan as one of the low and middle income countries, and you can say underdeveloped country, ranked fourth with the greatest number of preterm births globally. So despite this bleak scenario, all hope is not lost. So I believe that everyone has a role to play towards the prevention of preterm births and improve the care of premature babies. So with this objective in mind, I supervise the work collaborated with the Dow University of Health Sciences and Muhammad Ali Jinnah University. And so I supervise this work with my student Muhammad Bilal Asmi, and this is about his thesis research work. Okay, so you can already read the abstract on the GLBIO website. So let me take you to the methodology first. So we search and then extract 20 genes encoding 55 preterm birth, or you can say PDB proteins, from the NCBI, and out of 20 genes that encodes 55 PDB proteins, we extract genomic variants, or you can say single nucleotide polymorphisms or SNPs, from the ensemble genome browser. And then we filter exonic variants and then rare coding variants by downstream functional prediction tools, for example, SIFT, Polyphane, CAT, and so many other. And here the interesting fact that we use here the alpha frequency, because once you use 1000 Genome Project or NOMAD or EXAC or any other database, genome consortium databases, so mostly every sequence, every population genome frequency databases has its own frequency. Alpha database is a new effort from NCBI, and it aggregates all those frequencies in one allele frequency. For example, if you want to if you have interest in order to know the if you have a genomic variant, and if you want to know the allele frequency, the aggregated allele frequency in all population genomic resources or databases, then you have an alpha frequency. So then we filter all these variants on the basis of their alpha frequency, and we focused on South Asian population. And then we further filtered the database, and then we came up to 74 rare variants that were stored in just 4 genes. Okay, and then out of 4 genes, we checked that what would be those 4 genes which were highly expressed in female reproductive tissues, because we are interested in those genes that are highly expressed in vagina, in cervix, in myometrium, in uterus. So we checked all these, we checked the expression of all these 4 genes in GTEx databases, and along with we also checked the protein structural coverage, because we want to check that whether these proteins have their structures or not. Okay, so after having GTEx and protein structural coverage, we for this work, although it's an ongoing work, we are not stop on just one protein, but right here for this poster, we select calpain gene, more specifically CNN1 isoform 2 gene. So we just homology, we made a structure of this by the technique of homology modeling, and then we perform blind docking in order to detect its cavity site and then active site identification. So this active site identification is further supported by Pooch meta server as well. So after the selection of drug molecules from the drug bank, and after the selection of after the blind docking of progesterone, one of the highly used, you can say, inhibitor drug to stop the premature babies, or to stop the process of preterm birth. We use hydroxyquinone to have cavity guided docking, and then the identification of estimation of potential therapeutic intervening targets in order to prevent or stop the molecular process or physiology of preterm birth. So this is our methodology. And then so this is the structural coverage data of all these 4 genes. So number 1 gene is the calpain 1, although we have 3 other genes. For example, we have sleep guidance ligand 2, and then we have IQ motif containing 2, and then we have collapse collagen type as well. Okay. So as you can see, in CNN1 we filtered exonic variants 55. Silhouette, we have a lot of genes to a lot of genetic variants, 267. And the IQ gap, in IQ gap 2 we have 25, and collagen has 112. So, but if you talk about the pathogenic variants, so we focused on CNN1 because it is highly expressed in female reproductive tissue as per GTEx database, and it has high percentage coverage as far as its template is concerned. So its template is homology calpain domain, named as in PDB database, it is known as 1WYP. So this is the homology model of CNN1 right in front of your computer screen. And as we used progesterone, so once we docked progesterone onto the WYP, the template structure, the calpain homology domain, so it has energetic estimation of -6.4 with all these preferred amino acid interactions. So we use the same proton on the homology model of CNN1 isoform 2, and then we came up almost close and energetic estimation of -6.5 kilocalorie per mole. So the preferred amino acids interactions are a bit different from 1WYP because 1WYP is the CNN1 isoform 1, while we are working on CNN1 isoform 2. So I named it as transcript level homology modeling. So once you dock so once you have different compounds, for example, we have some preterm birth drug selection from drug bank, so they are total 23 numbers. And once we selected only approved investigational and experimental small molecules, so it come up to 17. And for example, those drugs that are involved in preterm birth management, we came up to the 7. And then all those drugs that passes Lipinski rule of 5. So we have 5 compounds. So we dock 5 compounds. So we have allosterol, retrosibin, ritodrine, terbutaline, and hydroxyprogesterone caprate, and these are their binding energies. So one of the most promising one is retrosibin, and if you see the molecular interactions of retrosibin, is it is highly hydrophobic in nature, while we have some interaction as well with bond 2.8 for angstrom bond of lysine 123. It's epsilon atom along with the nitrogen atom of retrosibin. Also, it has so many nitrogen atoms, but one of the nitrogen atoms which we named here as N2. So, I did not include the conclusion because this is an ongoing work. So we are still working further on 2 different proteins. For example, we are working on IQ-GAP2. We have the data, but we are still figuring out how to sort out the progesterone and its docking stuff. And then the next one is silhouette. We are not interested to for the homology modeling of collagen because it's more than 2000 amino acid residues, proteins so big, and there's no template. So we are just focusing on CNN1, silhouette2, and IQ-GAP2. So you can visit my poster at GitLab's Bioinformatics conference, this showcase virtual platform. So you can find my poster here as well, and you can find our poster over here. And I will be available on the 11th, 12th, and 13th May at the poster session timings. So I will be there to answer your questions, to have some more discussions, networking, and collaborations. Thank you very much. See you all there.